and WWP Genetics, we're thrilled to have you join us for another engaging and informative video. Today we will be discussing cannabis genetics and tailored strains, unlocking the potential of precision breeding. The evolution of cannabis from a largely underground and stigmatized plant to a subject of serious scientific inquiry has sparked a renaissance in botanical research, particularly in the field of cannabis genetics. The ability to tailor cannabis strains for specific needs, be they therapeutic, recreational, or agricultural, is no longer confined to the realm of anecdotal breeding. Instead, it is now rooted in advanced genetic analysis, high-throughput chemical profiling, and precise phenotypic selection. This movement toward customization represents a monumental shift, aligning cannabis cultivation with the standards and rigor of pharmaceutical development and modern plant science. Cannabis is a genetically diverse species, with a complex diploid genome that contains an estimated 800 million base pairs. This genetic diversity is reflected in its vast range of observable traits, or phenotypes, which include everything from plant height and leaf shape to flowering time and cannabinoid content. Unlike many other crops that have undergone intensive domestication and genetic narrowing, cannabis remains a rich pool of untapped genetic potential due to centuries of clandestine breeding, global dispersion, and minimal formal research until recently. With the advent of genome sequencing and the identification of specific gene loci associated with cannabinoid biosynthesis, breeders now have the ability to link genotype to chemotype, the chemical expression of the plant, including its production of cannabinoids, terpenes, flavonoids, and other secondary metabolites. Central to the tailoring of cannabis strains is the distinction between the outdated classification system of indica, sativa, and hybrid, and the more scientifically accurate concept of chemovars. While indica and sativa historically describe plant morphology and geographic origin, they offer little predictive value when it comes to pharmacological effects. Chemovars, on the other hand, categorize cannabis based on its unique chemical fingerprint. Advances in analytical chemistry, particularly gas chromatography, GC, liquid chromatography, LC, and mass spectrometry, MS, allow researchers to accurately profile the concentrations of dozens of active compounds within a plant. These compounds interact in a complex, synergistic manner often referred to as the entourage effect, where the presence of terpenes and minor cannabinoids can significantly modulate the effects of primary cannabinoids like THC and CBD. The tailoring of cannabis genetics for specific outcomes begins with intentional breeding programs that aim to isolate and stabilize desirable traits. These traits can range from high THC content for recreational use to elevated levels of minor cannabinoids like CBG, cannabigerol, CBC, cannabichromene, or THCV, tetrahydrocannabivarin, for targeted medical applications. Breeding strategies may involve hybridizing two parent strains with complementary chemical profiles, then selecting progeny that express the desired combination of traits. However, due to the polygenic nature of cannabinoid synthesis and the influence of epigenetic factors, considerable variability can exist even within a single generation. Therefore, large-scale phenotype hunting, growing out and assessing hundreds or even thousands of seedlings, is a necessary step in identifying elite individuals with the precise characteristics sought by the breeder. Once a promising phenotype is discovered, it can be stabilized through various methods. Cloning or vegetative propagation allows for the exact replication of the plant's genetic material, ensuring consistent chemical output across crops. For seed production, breeders often employ inbreeding techniques such as selfing, self-pollination, or backcrossing, breeding offspring back to one of the parent strains to reinforce trait stability. Recent advances in molecular breeding tools such as marker-assisted selection, MRs, enable breeders to identify genetic markers associated with traits like THC synthase expression or resistance to powdery mildew, expediting the breeding process by allowing early-stage seedlings to be selected or culled before full maturity. In the medical cannabis sector, this precision is being used to create strains with highly specific therapeutic profiles. For example, strains high in CBD and devoid of psychoactive THC are being bred for pediatric epilepsy and neurological disorders, as demonstrated by the success of cultivars like Charlotte's Web. Other strains are being optimized for conditions such as chronic pain, anxiety, insomnia, multiple sclerosis, and cancer-related symptoms. For instance, strains with a balanced ratio of THC to CBD can provide analgesic effects without overwhelming psychoactivity, while those high in terpenes like beta-caryophyllene and linalool may enhance anti-inflammatory or anxiolytic responses. The ability to breed strains that not only target specific symptoms, but also mitigate undesirable side effects has transformed cannabis into a truly personalized medicine. Beyond medicinal applications, genetic tailoring also extends to optimizing the plant for different growing environments and operational models. Indoor growers, for example, may prioritize compact plant architecture, fast flowering times, and high bud density, while outdoor farmers may focus on disease resistance, drought tolerance, and large yield potential. Greenhouse operators often seek photoperiod-sensitive strains that synchronize well with light deprivation systems and automated climate controls. In response to these needs, breeders are developing strains with targeted agronomic traits, including those that flower quickly, shortening crop cycles, resist common pathogens like Botrytis cinerea, and thrive under specific nutrient regimens. Auto-flowering cannabis strains represent a striking example of genetic innovation tailored for convenience and efficiency. Derived from Cannabis ruderalis, a subspecies native to Central and Eastern Europe, auto-flowering strains begin flowering based on age rather than photoperiod. This trait has been introgressed into high THC or high CBD cultivars through successive breeding, creating fast, hardy plants that are ideal for novice growers or those operating in challenging climates. Similarly, feminized seeds, produced by inducing female plants to generate pollen, ensure that nearly all resulting plants are female, thereby eliminating the risk of male pollination and seed production in commercial flower crops. As cannabis breeding continues to intersect with modern biotechnology, the future may see even greater precision in genetic tailoring through techniques like CRISPR-Cas9 gene editing. These tools allow scientists to directly modify genes responsible for key traits, potentially accelerating the development of new cultivars without the lengthy generational cycles required by traditional breeding. For instance, researchers could knock out genes that lead to unwanted metabolites or insert genes that increase cannabinoid production. However, these methods are currently limited by regulatory hurdles and ethical debates surrounding genetically modified organisms (GMOs), particularly in the cannabis industry where natural and organic cultivation are highly valued by consumers. In addition to laboratory science, data-driven feedback loops involving consumer and patient experiences play a critical role in refining cannabis genetics. Digital platforms and mobile apps now collect real-time user data on strain efficacy, side effects, and preferred uses. This information can inform breeding priorities and help producers match their cultivars to market demand with greater accuracy. Such integration of bioinformatics, consumer research, and plant science is gradually transforming cannabis breeding from a largely empirical art into a multidisciplinary science. In conclusion, the field of cannabis genetics has progressed from informal cross-pollination to a sophisticated practice of precision breeding and chemovar optimization. Whether for medical patients seeking symptom relief without intoxication, recreational users desiring specific psychoactive experiences, or cultivators aiming to maximize yield and disease resistance, tailored cannabis strains now offer solutions that were unimaginable just a few decades ago. As the industry matures and regulations evolve, we can expect continued advances in genetic research, strain development, and plant biotechnology, further solidifying cannabis as a plant of profound versatility and significance in both science and society. We hope you found today's video informative. Don't forget to subscribe, like, comment, and share to stay updated on our latest content. For more exciting insights, be sure to check out our brother channel at WWP Logistics. See you in the next video.